welcome to the Radiation Research Society podcast. We are filming today here from the conference in Las Vegas, and I have with me Claire Vanpouille-Box from NYU School of Medicine, and I'm Allison Burrell, and I'm here to talk about your work and which award did you win this conference?
I got the honor to receive the Marie Curie Award, and I was very surprised to get awarded because nobody expected that. So thanks again for the society for selecting me as the Marie Curie Award recipient this year.
Yeah, congratulations on that, and we'd all love to hear more about your work.
Yeah, sure. So basically, we try to see if the type of radiation regimen can influence the generation of an in situ tumor vaccine. So because our group have previously shown that radiation is capable of generating an immune response, an in situ vaccine, we, depending on the radiation regimen we were using, we were comparing actually a single fraction of radiation of 20 gray versus 3 times 8, an hyperfractionation regimen 1. And we first actually look at gene expression patterns after 24 hours of radiation, and looks like we got actually similar results that were obtained in Professor Coleman's lab, showing that we have this type 1 interferon induction after multifraction that was not observed after a single fraction. So we were wondering, because we knew that this multifraction regimen was better at synergizing with immune checkpoint inhibitors, if there was kind of something that we didn't know about. So we go further, obviously, and we look, because actually, which was very surprising to us, is to see a type 1 interferon response and not the interferon gamma response, which is different. So we look at literature to see if there was a relationship between interferon type 1 and the immune system whatsoever, and we find a very preserved, I would say, biological pathway in which interferon type 1 activate dendritic cells maturation. So we look at maturation of dendritic cells after multifraction versus 20 gray, and obviously we find a better recruitment of those cells within the tumors that were also more activated. So we were like, okay, that's maybe what is happening. And so we go further because, we were like, okay, what is... Because you have to know that type 1 interferon is very specific to a virus infection. It's usually never seen in any other cases. So something which was very exciting, but also deceiving, because we were like, okay, how can I make the link with radiotherapy? Right. And so we found this very interesting publication by Chen Labs in University of Texas, in which he described kind of new, I would say, pathway that is mainly mediated by cGAS and STING, in which DNA is sensed by cGAS to activate STING and thus produce interferon type 1. But this was only described in viruses condition. So because radiation is inducing DNA damage, obviously we were thinking, okay, maybe there is a link. So we look at that, and turns out that it was the case.
So this is radiation, a hyperfractionation dose that is mimicking a viral infection.
Yeah, that's the conclusion mainly of the talk. Yeah.
That's very interesting. And what type of cancer cells are you using?
So we did all the experiment on the TSA mouse breast cancer, which is a syngeneic, so immunocompetent mouse model. So I have to expand and do this experiment to other tumor types, but we know for sure that from our collaboration with Professor Coleman and Dr. Aren Cavalier from NIH, that they obtained actually the same kind of gene signature after multifraction of human prostate cell cancer.
Okay.
So it's kind of suggesting that we can see the same effect in other tumor types.
Now, in the clinic, hyperfractionation is already being used.
Yeah, for breast cancer, 5 times 6 as already is used usually.
Do you think it would be possible to look at a patient population and just with blood samples and evaluate if there is certain like an increase in the immune system, or is it some time points that or endpoints that they're already looking at?
I know that from a case report that Sylvia Formenti actually presented during the debate session from the RRS meeting.
Yes.
That so she had a patient metastatic for non-small cell lung cancer with multiple metastases in the bone, lungs, and liver. And when she combined fractionation radiotherapy of 5 times 6 with immune checkpoint inhibitor anti-CTLA4, they observed abscopal effects all over the body 6 months after the treatment, which was very fast to me.
Wow.
And this patient is still alive right now, 2 years later. So, which was very interesting. I cannot tell you if we can really see type 1 interferon in patients, but what we know from this case is that, from a biopsy, while he was responding, we saw nice infiltration of lymphocytes, suggesting that this was mediated by the immune system, but I cannot say more.
Yeah. Well, what was the time point after the fractionation in your mouse model that you looked at this?
The type 1 interferon.
Yeah.
So we looked at 24 hours most of the time, because we really want to look at the direct effect of radiation therapy. We didn't want to see, because sometimes you have a small effect that will
Right.
give another huge effect. So we really wanted to focus on that, and this is why I looked at 24 hours after the last radiation exposure. Yeah.
Well, it might be interesting to look at something. I mean, I know it's difficult in mice because they have a shorter lifespan, but you're saying this patient that Sylvia was working with didn't see that effect, that abscopal effect, until 6 months after the initial treatment. So do you think it was maybe a prolonged increase in the immune response that carried over that you could look at in the mouse as well? Like if you, instead of just looking at the acute response, is that something you would want to look at?
Actually,
is like a longer term, like are you still seeing, the immune response, I don't know, a month out or something?
Yeah, but actually we already look at that.
Oh, you did?
Yeah, because I think it was in 2009 in Clinical Cancer Research, our group published that CTLA4 was more synergistic when combined with 20 gray, a segment dose of 20 gray, or multifraction of 3 times 8. And this is why we actually look at those two radiation regimens, because we knew that 3 times 8 was generating abscopal effects in the mouse. So that we knew. So we just didn't know. We just wanted to know actually why 20 was not that good compared to 3 times 8. Because the control of the irradiated tumors was the same. It's just the control of the systemic response that was different.
Okay.
So, yeah. We just wanted to decipher why actually. And I think this is the question that everybody is asking. So this is why we did that work. Yeah.
Thank you.
You're welcome.
All right. Thank you guys for joining us here in Las Vegas for the podcast. And thank you again, Claire, for speaking with us. Have a great day.